All right. Today we're going to talk about crossing monkey flowers, doing controlled crosses in the yellow monkey flower, Mimulus guttatus. This beautiful little plant is a model for scientific research, and particularly for genetics. All right. One of the things you have to do in genetics is perform controlled crosses, where you transfer gametes from one organism to another. We're going to work through the procedures to do this in yellow monkey flower. What we have here is an artist's representation, a side view of a dissected monkey flower. This is the calyx, which is this green tissue here. And what you can see extending through the middle of the calyx are the female reproductive parts. This is known as the pistil. At the base of the pistil are the ovules, the eggs of the plant. And this, the style, is what will transmit or confer pollen to fertilize the ovules. So in doing a controlled cross, we will transfer pollen to the stigmatic surface here. And over the span of a couple days, pollen tubes will grow down the style and fertilize ovules at the base of the flower. In yellow monkey flower, it takes about 2 weeks, 2 to 3 weeks, between hand pollination and when you can collect seeds. One of the nice features about yellow monkey flower is the ease at which you can do crosses and the speed at which you can go from seed to seed. In other words, it has a short generation time. Any questions?
Do all plants have the capacity to self?
That's an excellent question, young man. It turns out that the capacity to self-fertilize varies among monkey flower genotypes. Okay? Self-fertilization can occur within a flower when the anthers, which are affixed to the corolla is the yellow tissue, what we often think of as the petals, the male parts, or the anthers, which have the pollen, if they transfer pollen to the stigmatic surface, you can produce what are called selfed progeny. All right, progeny derived by sexual reproduction, but within a single plant. Now, some plants do this quite naturally. In fact, some species of plants do this habitually. They'll produce almost all of their seeds by selfing. Yellow monkey flower is a mixed mating species. In other words, it routinely produces some of its offspring by outcrossing and some by selfing. This population of yellow monkey flower is predominantly outcrossing. All right, so most plants will produce outcross progeny most of the time. But we do see that some genotypes from this population are quite efficient at selfing. Others, not so good.
How do you prevent cross-contamination when crossing and selfing many plants?
What do you mean by cross-contamination?
I mean, how do you know that you got the right pollen in the right plant?
Well, by carefully cleaning our forceps between controlled crosses. All right. Later we'll go through, at the bench, the actual procedures using forceps to transfer pollen.
How can you tell that you successfully pollinated?
Well, what we're going to see when we do a hand pollination is that the stigmatic surface will close following contact. All right? In other words, after we transfer pollen to the stigmatic surface, it'll close. Now, if we don't successfully convey viable pollen to the stigmatic surface, what will happen is this stigma will reopen. All right? And that's an indicator, at least in our population of yellow monkey flower, that your cross has failed. Of course, the fact that the stigma has reopened allows you to try again.
Your plants look like they have lots of flowers. How can you tell which flowers you've crossed or self-pollinated?
Well, what you can do is after you do a cross-pollination, you'll put a small dab of paint on the calyx. Now, you have to imagine that at the moment because I don't have paint, but you can place paint on the calyx and that will mark it as a successful cross. It can also indicate who the father in the cross is. It's easy to tell who mom is because mom is the plant you transferred pollen to. All All right, dad can be a bit of a mystery, but you can code dad either with the color of the paint or by clipping the male plant to the female plant. The other indication of a successful cross is that as seeds begin to mature, the ovary will inflate and the entire calyx will become kind of larger and spherical. If the cross fails, it will tend to shrivel and not produce the kind of fruit that we see, the product of a successful cross. Yes, young man.
How can you tell when seeds are produced and ready to come off the plant?
Well, the calyx, once inflated, will often kind of leach of its green photosynthetic tissue and become kind of brown and brittle and transparent. And you will see the ovary, which is swelled, crack open and release seeds. All right, so if we have an inflated pod, all right, you'll see the seeds collecting at the base of the calyx. And as I said, it'll become transparent, or at least partially transparent, by the time the fruit is mature.
Is there a difference in pollen quality between selfing and crossing plants?
I wouldn't say that the pollen quality is different. The quality of the zygotes produced may well be different. All right? The zygote is the diploid seed, the product of pollen and ovule. All right? And if you combine pollen and ovule from the same plant, that's what we call inbred progeny, and they might well be inferior in terms of survival or reproductive success than outbred plants. Any final questions? All right, then let's progress to the bench.